Section 1. Introduction. Over the last decade, the transformative concepts of information processing and propagation in the molecular domain have dramatically reshaped the frontiers of communication and networking research, particularly within biomedicine. As a result, advancements in nanotechnology and biotechnology have spurred the development of wearable and implantable devices, featuring enhanced biocompatibility and operational autonomy. These devices are designed to sense, process, control, and exchange body health parameters continuously, accurately, and in real time, thus supporting remote monitoring under the umbrella of the Internet of Bio-NanoThings, IoBNT. This paradigm promises to revolutionize the way we understand and interact with the body's biological dynamics, paving the way for breakthrough biomedical solutions to previously unsolvable clinical challenges. IoBNT envisages a heterogeneous network of electronic and biological devices, situated both inside and outside the body, as illustrated in Fig. 1. These devices communicate using a variety of methods, including electromagnetic waves, electrical and mechanical stimulation, and molecular communication, MC. The electronic segment includes devices like brain implants, pacemakers, and smartwatches, while the biological segment consists of both naturally manipulated cells and tissues, or synthetically engineered entities such as immune system cells, gut microbes, and artificial corneas. Figure 1. The Internet of Bio-NanoThings paradigm. Developing the IoBNT communication network infrastructure involves several critical prerequisites: accurate and predictable models of communication and networking parameters, minimal interference with natural body functions to avoid psychological or physical discomfort, and interconnection of heterogeneous devices, electronic and molecular, in a manner that is accessible externally in a minimally invasive way. These requirements challenge traditional telecommunications approaches, especially for establishing intra-body IoBNT links. This paper proposes a conceptual framework to exploit natural biological communication systems within the human body to connect wearable and implantable devices. Our focus is on the Microbiome-Gut-Brain Axis, MGBA, where electrical signals are converted into molecular signals affecting gut microbial communities, and vice versa. This synergy relies on natural communication links and interfaces within the nervous, endocrine, and immune systems. The holistic approach of the MGBA integrates electrical and molecular communication and their interfaces. The external accessibility through the alimentary canal, coupled with the genetic programmability of microbial cells, underscores the potential of this system within the IoBNT framework. A direct connection to the MGBA provides extensive data on the health of the central and autonomous nervous systems, as well as the gut. This article explores the use of the MGBA as an IoBNT communication network infrastructure to transmit and receive information to and from electronic and biological devices, as depicted in Fig. 2. The envisioned infrastructure is also designed to interact with the external environment via dedicated molecular, alimentary canal, and electrical, wireless transfer through skin interfaces. Figure 2. Microbiome-Gut-Brain Axis IoBNT communication network infrastructure. To achieve this, we present a methodology that includes both analytical and experimental approaches. The analytical effort builds on neuroscience and bioinformatics to model the propagation of device-sourced information through biological tissues, covering electrical communications in the gut muscle, enteric, and autonomic systems. Molecular communications involving biological devices and natural gut microbes via hormones, neurotransmitters, and metabolites, and the interface between these electrical and molecular communications in the gut and central nervous system. The experimental approach involves designing an integrated network probe device for in vitro use in a novel gut-on-a-chip system that co-cultures by an implantable version for in vivo data collection in laboratory rats. This paper is structured as follows. Sec 2 details the derivation of analytical models for device-to-device communications from biochemical processes within the MGBA. In Sec 3, we outline the design of devices and the empirical data collection methodology to augment the analytical channel models in both in vivo and in vitro settings. In Sec Voit, we discuss the primary elements of an IoBNT network infrastructure based on MGBA channel models, and describe a simulation environment to support network design. Finally, in SESUV, we conclude our findings. Due to its controllability, GOAC provides a perfect experimental testbed to study the MGBA communication channels where the individual communication links can be isolated and tested. Also, this platform limits noise and variability among the individuals compared to an in vivo setting, which helps in the definition of general models. Moreover, this in vitro platform will be also utilized to test the devices and interfaces designed considering the criteria described in SEC 3A to assure accurate operations and biocompatibility before conducting in vivo experiments. In this controlled environment, by tuning system parameters to extremes, limits of operation without damaging tissues and altering the microbial balance will be examined. While GOAC systems can achieve control over specific individual aspects of the tissue environment, existing examples are still relatively simple compared to the actual gut ecosystem. To create a GOAC more reflective of the physiological function under test is a challenge that needs to be addressed. Other challenges include culturing human intestinal cells, living microbiota and enteric nerves in the same chip while maintaining cell viability. Following the in vitro experiments, refined channel models for communications over the MGBA and tested devices capable of interfacing with the MGBA will be integrated to be tested further in an in vivo experimental platform. In order to capture other unexpected properties of the actual body environment and dynamically measure communication parameters, in vivo experiments should be on animal models. In preclinical studies, researchers prefer to use small animal models such as rodents because of lower cost, rapid growth, ease of maintenance, and similarity of their biological and behavioral characteristics to those of humans. Here, we describe our methodology based on rat models, which are very suitable to observe the overall effects on a live.